Christoph Lippert. I'm a professor at the Hasso-Plattner Institute in Potsdam, Germany. Yeah, I work on what you broadly could say methodology for multimodal health data, which exists in mass, as you all know, and requires some methods development. So we spend a lot of time doing that, but also do this always in the context of studies and with partners on the biomedical side. The goal, of course, is generally to leverage this data to get closer and closer to understanding diseases, to detect them earlier, to maybe be able to intercept disease processes as they develop in order to get people better chances for managing disease, treating earlier, and so on. Yeah, so today I'll talk about a problem in genetics. As you know, genetics traditionally has been done in a way that you would collect the genotypes of people, you collect their phenotype, and the phenotype that is easiest to collect is the distance between head to toe, so the height of people. And then you essentially correlate these two things and you find associations such as you see in this picture showing a nice association between the sex chromosomes and the distance between head to toe. Yeah, has led to a lot of associations with diseases being found, and they have been recorded by the NIH and by the EBI in these databases, we are working in a consortium since, so this is the last year of this project, unfortunately, because it was a really productive undertaking, where we want to take all of these known associations in order to characterize disease risk on the one side, but also leverage from these large biobanks and clinical data sets that we in the consortium have access to, integrate these with other types of data. So think about electronic health records, think about imaging, and so on. Everything that you could think of. So it's a really multimodal problem. So it's on the core are genetic risk scores, polygenic scores, but also try to combine genetics and estimate risks from other things. So polygenic risk, I mean, if you look at what a single variant typically does on these complex traits, it's next to nothing usually. However, polygenic risk is actually quite powerful. It can characterize risk quite well, and this is an example from one of the papers that came out of this consortium, where we look at the risk for type 2 diabetes, and we essentially stratify people by the level of risk. So you see here the blue line. This would be in this case, women that lie in the top percentile of risk based on their polygenic risk score. And here you see sort of the roughly median range. And here you see the people in the lowest percentile of risk. And you see that if you look at the incidences, that's actually what's plotted on the incidence rates. That's what's plotted on the y-axis versus the age. That people who are in this very high risk category, they develop type 2 diabetes, tend to develop it earlier, and also much more in total. So by the age of 80, about 70% of these people will actually develop type 2 diabetes. So you can almost surely predict it from their polygenic risk. And for males, this looks pretty similar. And what you see also here is this red line. This is sort of the recommended age for screening for type 2 diabetes. at age 45, and it's sort of essentially age 45 would be here, but that's sort of the risk that the median person would have at that age would be about 8%, and you can see if you are in the highest percentile, they reach this risk already in the early 20s. So there is the potential to use this data to stratify screening better, make it more efficient. And like on the other side, you see the people on the lowest percentile, they actually never reach this risk that the average person would reach with the age 45. So one of the things that we did in this consortium was to look at sort of different methods to estimate polygenic risk first. And because we had access to people working with all these different biobanks, we have also evaluated all of these methods for different diseases in the various biobanks. And what we saw is that generally differences in biobanks tend to be more important than differences in these methods. They're essentially all variants of linear models, so I guess that's no surprise. That's true as long as you stay within European population. If as soon as you go look at people from different ancestries, everything breaks down and they essentially all don't work very well anymore. As you can see here, if you look at the difference, so this is again risk for type 2 diabetes, but this is true across all of the different traits. You see that the quality of prediction drops down drastically if you go to non-Europeans. There's also a different exception. It's not on the slide. Is that if you look at immune-related traits, there are actually differences between methods matter a lot. However, that's mostly related to how they process the MHC region, because some ignore it completely because it's kind of complicated. Surprise, surprise, they predict much worse than others that do. And amongst that do, they have very different ways to tackle this complex regions, and it matters much more. So there's actually, this told me there's quite an opportunity to do risk prediction for immune-related diseases, just spending some time of better characterizing the MHC regions with better bioinformatics. So overall, the take-home of this big effort, essentially taking all of these methods on the different biobanks, different diseases, was differences between methods are pretty negligible compared to the differences between the biobanks and ancestries. And I will also come back to these differences between biobanks. I mean, one problem is that phenotype definitions tend to vary a little bit. Then you have different diagnosis standards and so on. So it's quite variable, and originally we were surprised, but however, it's maybe not so surprised, but maybe there's some solutions also to that. So that was sort of the baseline that we built on in this consortium, however, main goal was to integrate this with other kind of information, so to go beyond these very simple traits towards utilizing all kind of different data types that we can. And turns out that that's not so easy. This really requires a lot of new methods development. So we started out very simple and worked on problems like thinking about, okay, how can we determine the difference between two sets of images. For example, you have brain images of people with an ApoE3 mutation and allele, and images of people with an ApoE4 allele. So so they have different risk for getting Alzheimer's. Then how can we do this? Essentially, what we're dealing here is a so-called two-sample test. So you have samples from some distribution P that's our ApoE3 samples, and samples from distribution Q. These are our E4 samples, and you're asking the statistical questions, are P and Q equal or not? Yeah. Now, traditionally, if you already knew what you're dealing with, so if you already knew that they have a different risk for Alzheimer's, you would probably look for a biomarker for Alzheimer's in these images, right? I mean, you would look for example, the ventricle size. So if you get dementia, the ventricles tend to enlarge. So you would then again be in this very simple setting where you essentially do look for the differences in mean between the ventricle sizes in these two groups. Yeah? So, however, it's kind of not scalable. So what you could do next is, instead of determining this all manually, you could train a neural network to do a regression of ventricle size, so that is also, of course, feasible. Yeah? So you estimate the volumes of the ventricles, and then you have your number, and you can do your traditional statistics. Now, once you already have some neural networks that work on images, you can actually do a different trick, and that's actually much more elegant, because not always, I mean, if we already know what the biomarker is, we don't need to do scientific discovery. What we're really after is finding potentially new biomarkers, finding things in these images that we don't know. So what you really want is to make as little assumptions as possible. And the fact that neural networks are powerful feature extractors, you can use and work with these extracted features directly. So essentially what this boils down to is to work with vector embeddings. And it turns out there's in the end, very simple two-sample test that you could do is to take these vector embeddings and compare the average embedding of the one group and the average embedding of the other group, and compute the distance between these mean vectors. And the norm of this difference vector turns out this is a non-parametric two-sample statistic. Yeah? And it covers with very mild assumptions on these distributions any kind of differences in these two groups of images. And for machine learning people, it's a known statistic as the MMD statistic, so maximum mean discrepancy between X and Y. And of course, we are applying this to these ApoE3 and ApoE4 samples. We also detect that they're indeed not identical, these two groups. Now, of course, if we want to do genetics, I mean, this wasn't a genetic example, but it was a very simple one because we had two alleles, but we want to be a little bit more general than that. But the trick that we're after is always the same, is we want to avoid, taking these complex things and either binarizing them very crudely. So think about binarizing brain MRIs or extracting a single number, but rather use neural networks to avoid this information loss, to essentially keep around as much of the information in the images as possible. Now, of course, that two-sample test is somewhat too simple to do genome-wide association studies. So, because what you typically do in a genome-wide association study is you actually have three sets of variables. So you have X, that might be your genetics, you have Y, the phenotype, and then typically you have a set of covariates that you want to adjust for Z. And typically what you do is then test for conditional independence between X and Y, conditioning on the covariates. So really, we are looking for conditional independence tests. So we then started to work on conditional independence tests. Now, turns out was kind of hopeless from the beginning, because there's a famous result by Shah and Peters, who showed that this is a fundamentally impossible problem, because you either, so if you're truly non-parametric, then that means also that your modeling of Z has to be very flexible, which means that you will have zero power, because it will be able to explain away all sort of dependencies between X and Y. Or if you actually sort of don't model Z very flexible, if you're non-parametric, you cannot control the type 1 error, because you cannot guarantee that any sort of dependency between X and Y is actually controlled for. So, okay, we knew already that's not possible, so we look for many simplified assumptions, and we've developed a bunch of tests that do so, and and you can find them compared in this paper here by Marco Sinnmacher. And a very simple thing to do in the context of GWAS is to just assume that once you are in this embedding space, everything is linear. That's of course not non-parametric, but that's typically people do conditional independence tests in these in linear models, anyways, in the GWAS setting. So it's probably good enough. So turns out this works quite well. We have so if you have been here 2 years ago, you actually saw me present the results of the first GWAS that we did with this method. That's on retinal fundus images. Let me briefly review what the method does. So essentially, you use a already trained neural network. Yeah. So you somehow need the feature extractor, and this, like where you get this trained neural network from, you're pretty free. The only thing that you shouldn't do is use the images that you actually want to test on, because you shouldn't use the data twice. So you cannot train the neural network and then test on the same images. That will mess up your statistics. So rather download a neural network that has been trained on ImageNet. Even if you're dealing with retinal fundus images, it's better than using this one data set of retinal fundus images that you have. Another alternative would be to split your data set in two. So use one to train the neural network, and the other one do the testing. However, then you have quite a hit of power, because you are reducing your sample size for testing. So the strategy that we then chose instead was actually to look for other data sets of retinal fundus images. There are plenty on the web, and train a neural network based on those. And that's essentially you do some sort of transfer learning. So you have, and they don't help us in terms of power anyways, because they don't come with genetics. So we can use them to train the neural network. So the output of this is a neural network. Could be a convolutional neural network, could be a different neural network. Overall, this method is quite flexible, because in the end, what you're really interested in is just the embedding layer, or an embedding layer, some hidden layer protection of the neural network. And then you might want to further condense this, for example, using a principal components analysis to reduce the dimensionality of this embedding layer further. And then you do your linear modeling on this condensed feature vector representation to do association testing, and that gives you your GWAS results. So that was what we did there. Since then, we have applied this to a more challenging type of data. So regional fundus images are nice because they're just 2D images. They look very similar to the pictures that you take with your cell phone. If you look at MRIs, they're not quite that similar because they're volumetric. So, so last year we did a TransferGWAS of these brain MRIs in the UK Biobank. We again wanted to try out this use of ImageNet. In that case, ImageNet of course are 2D images. We just applied them slice by slice to the brain MRIs and then averaged the representation that we got from the different slices. But we also trained a 3D architecture on samples from the Alzheimer's Disease Neuroimaging Initiative. So we have 2 sets of embeddings. And now we can do an analysis with these 2 and combine them or compare them to see what we get. This is the type of Manhattan plot that you get if you start with the ImageNet pre-trained embeddings. So remember, we used cats images and dogs images and whatever to, in the end, do genetics with brain MRIs. And turns out we get a bunch of associations. Now I will come back to what these associations are. Is that a question?
Yes, very short one. I don't want to interrupt. What is this Manhattan plot of? What is the phenotype?
Yeah, so it's okay. What is the phenotype? So let me quickly explain the plot. So on the x-axis we have the position in the genome, tiled by the number of the chromosome. On the y-axis, we have for a given position then the strength of association. You see that there are many of these associations that are sort of clusters. So loci, they are in LD with each other. They tend to be associated together. And in this case, the phenotype is brain MRIs, embeddings of brain MRIs. That's the. So there's an association between some patterns in the images and the corresponding locus in the genome.
Patterns of brain MRI.
Patterns, so some variation in MRIs. Yeah.
This is done for each coordinate of the embedding separately, or?
No. Yeah, well, yes, we do it. We essentially combine the p-values for each. Yeah. So it's essentially a multivariate test. Yeah. So we reduce it, I think, in this case to 20 principal components, and then we compute the max.
So it's the existence of completely abstract patterns.
Correct. So like all the interpretation has to be done post hoc by looking at the individual loci. Yeah. So it's a kind of abstract phenotype. Here is the result if we use a neural network that has been pre-trained on other regional fundus images. In this case, it was data from a Kaggle competition to predict diabetic retinopathy. So it was a classification task. And yeah, of course, we find a lot of associations. But these phenotypes are kind of abstract. So how can we explain them? So we spent a lot of time essentially then implementing a post hoc sort of interpretation pipeline, where on the one side we would do, look at, okay, so these extracted embeddings, do they associate with any phenotypes in this cohort? So any known phenotypes that are reported in the UK Biobank anyways. And of course, for the associations that you find with these brain MRI representations, we can also look at these loci and see whether these loci have been reported associations before, by looking them up in the GWAS catalog. And the third thing that we did is to take now the set of associations that we get, these loci, and map them at the images directly. So instead of embedding the images first, we now do a voxel-wise test. So these MRIs, they're essentially, think of voxels as the volumetric equivalent to a pixel. So normal 2D images have pixels. Volumetric images have voxels. So it's like there's the VO for volume, right? So it's like a little cube, essentially. And you have different grayscale values in these cubes, and you're essentially looking for the association between the variation in the grayscale values in these loci. So first the GWAS. We especially found that the brain, the images, the association, the representations from ImageNet tend to be associated with general sort of growth-related traits. So everything that is sort of, general structure. Yeah? So, which can be explained from the fact that these filters, they are not at all sort of specific to brain images, right? So they essentially, these are very low-level patterns that extracted from these MRIs. So you see something like bone density and so on, right? I mean, these are mineral density, cortical thickness, and so on. And if we look at the associations of things related to ADNI, then we actually see much more specific associations to individual organs of the brain. Yeah. And that's also visible in the parametric mappings. So remember, we now take the associated variants and we test them against individual voxels, and then of course we can look up where the significant associations map to. Yeah? So each of these dark dots would be an association to one of these things. Yeah, I won't go into detail here. Feel free to look at the paper. Similarly, I mean, similarly, if you look at genetic correlations with other traits, the results are similar. So generally, the embeddings from ImageNet tend to be associated with these very general growth-related traits, and the ADNI ones more specific, really to the brain. So let me summarize this part. So in order to associate images in a very general way to genetics, we work on these new statistical methods that can take complex high-dimensional data using neural networks. And the problem now is that the results, that the phenotypes themselves are because we don't extract any specific phenotypes, are not interpretable themselves. And we have to spend more time on post-hoc interpretation. So we also do a lot of work on XAI methods. So, however, I didn't include them in this talk here. So I have some time left, and I want to tell you about some other work that we do in Intervene, which is more related to electronic health records data and predicting, using the patient history of a person to predict sort of a future disease endpoint, and also leverage the fact that we have within Intervene access to electronic health records from different health systems. So the question here is, can we take a model that has been trained in one health system and evaluate it somewhere else? And the short answer is this doesn't work super easy because the the features that we find are actually quite different in distribution. So why is this? Well, for example, the same phenotype might be sort of like the same code, even if you use the same sort of coding scheme, such as here, for example, OMOP, which is a data exchange format. That is used both in the UK Biobank and here in this case in FinGen. Is also used at Mount Sinai. We see that for example, if we look at some upper respiratory infection, that the frequency of use of some very related term, so of acute upper respiratory infection in UK Biobank is much higher than in FinGen, but in FinGen we see a lot of just upper respiratory infection. so if you just work with these sort of one-hot encoded features, your correlation goes to 0 because these never match, right? So what can we do? And Matthias had a very simple idea, to use the fact that they are semantically quite similar and that there's a description for each of those codes. And so the idea was essentially to use an LLM encoder model to take not the code directly, but the description, text description of the code in order to get an encoded feature vector. And it turns out that they tend to be quite similar because they're semantically quite similar. So while the code, of course, is cryptic and not identical, the text encryption, the text description is quite similar, and you are able to map these codes. So the resulting model that we then used to train on one health system and evaluate on another health system in the end is then a very simple transformer model where you take the patient history. So you take all, actually you don't even need to feed sort of patient information into the LLM encoder, because what you just do is you go through your ontology, so through your coding scheme, and for every code that you have, you take the text description and you embed it. And then you get an embedding per code. And then essentially you treat the association between a code and this as a way to retrieve this embedding vector for your patient. So you just once encode all you embed all the codes, and then you essentially have something like word vectors for these codes that you can then use to train a transformer to predict the endpoints. And long story short, this works quite well. So what you see here is sort of these are 23 different disease endpoints, and we compared different embeddings. You see here a model that uses only demographics, and then one that uses random embeddings versus the large language model-based embeddings. In this case, it was based on GPT-4. So OpenAI embeddings, and consistently both evaluated within the same biobank, but also if you then take a model that has been trained on UK Biobank and evaluated on patients from Mount Sinai, there it improves still the prediction for every single endpoint that we tested. So this is quite nice. So these are two data sets that have been trained. These models have been trained on FinGen and evaluated on the UK Biobank and on Mount Sinai. Turns out you can do a bit better if you then also fine-tune a little bit on the cohort. But I don't show these results right now. But rather I want to show you another thing which is quite cool, because what we've seen so far was, training a model on an OMOP encoded dataset and testing in a different OMOP encoded dataset, which is hard enough. But what this also allows us is to completely ignore the type of encoding that you have, because you can even translate between different encodings with this. So here in this case, we compare a model that has been trained this has been trained on OMOP and evaluated on ICD-10. which is a different set of codes. So you wouldn't even, not just the frequencies change, but you don't even have the same codes. So we found that pretty cool. And yeah, here I would like to quit the talk, say thanks to my group, and take some questions.